Hello and welcome to Nikolay's genetics lessons, and today's question I got from one of my subscribers. Here's a question: why is there only one band appearing after double restriction enzyme digestion? And here's a problem statement. Currently I am using the pETDuet-1 vector 5420 base pairs for cloning. I used restriction enzymes BAM-H1 and SAL1 for restriction enzyme digestion and checked whether my vector was cut through the gel electrophoresis. In lane 4, I used both BAM-H1 and SAL1 to cut my vector. As you can see, only one band appeared on the gel, and I was wondering why one band appeared. Since I used two restriction enzymes, shouldn't there be two bands? So here's a lane 4 and we see only one band. So here's the logic. We have circular double-stranded DNA, which we call vector, and we cut it in one place with one restriction enzyme and in other place with different restriction enzyme. And as you see, we should get one band which is smaller, for example, and another one which is bigger. Or depending on the size, we can get two of the roughly the same size, or one can be much smaller, another can be much bigger. But we expect in the lane 4 to see two bands. So what happened here? The explanation of this gel electrophoresis is probably going to take me only 10 seconds, but I know that many students watch my videos, so I want to explain what this vector use it for and why we can sometimes see unpredicted picture like this one. So take a look at this vector. So first of all, pETDuet vector is a type of plasmid vector which is commonly used in molecular biology, research for expression of the recombinant proteins in bacterial systems, and particularly in E. coli. pETDuet vector is dual expression vector, meaning it allows for simultaneous expression of two different target proteins in the same host cell. Now let's talk about abbreviation, how this vector was named. The PET in the vector name refers to the T7 promoter-based expression system, which is widely used for high-level protein expression in E. coli. The T7 promoter is a strong promoter that can drive efficient transcription of the target genes, and as you see, here's a list of the sites for restriction in the nuclease, which integrated here, which we can cut with these restriction enzymes. The duet in the vector name indicates its unique feature of allowing two different genes to be expressed simultaneously. The vector contains two multiple cloning sites, or MCS, each with its own promoter and ribosome binding sites. These elements allow for independent expression and regulation of different genes or protein domains within the same host cell. So this is domain 1 and this is domain 2. What we use this vector for? What would be applications? The pETDuet vector is used in situations where researchers want to express multiple proteins that may have functional or structural interactions, or need to be co-expressed for proper folding or activity. In other words, it simplifies process by allowing both proteins to be expressed from a single plasmid construct, reducing the need for multiple vectors and potential complications associated with coordinating the expression. Now let me show you the sequence of this vector. Take a look. Here is a sequence of these two domains. What do we see here? Here is a T7 promoter 1 of the domain 1, and here is T7 promoter 2 of the second domain. We also see here ribosomal binding sites 1 and 2. Here we also see nucleotide sequence of these domains. So we are interested in domain 1 because both restriction enzyme recognition sites are in this domain. Do you remember the names? The first one was BAM-H1 and the second was SAL1, which is here. And underneath each name we see specific nucleotide sequence. The first recognition site for restriction enzyme would be cut between nucleotides G and G. The second one, restriction enzyme, is going to cut between nucleotides guanine and thymine. Now let's find how many nucleotides we have between these two cuts. I actually already counted and we have 31 nucleotides between these two cuts. This is going to be our smaller band. Now we can return to our problem. How big is our vector? It is 5420 base pairs. How big is our single band? As you see, it is double cut, lane number 4, and it is around this size. So, as you remember, the smaller fragment have to be 31 base pairs long. That means that the long one have to be 5389 base pairs. So why we don't see the second band? Say somewhere here. The answer is very simple. It's just so small we wouldn't be able to visualize it. Basically, when we have two cuts which is so close to each other, it's basically for us means one cut. So we are going to get one long molecule and another one is going to be so small that we just wouldn't be able to see it on this gel. So basically this line represents double cut, but they are so close to each other that I even cannot put another line, or I can put on top of this line. This second band is just too small. As I said earlier, I could explain why we do not see a second band here in just 10 seconds, but in this case I hope you wouldn't have so much fun. And this is all for today. Subscribe and see you in the next video. Goodbye.